Hey everyone, and welcome back. So, if you're gearing up for a genetics exam, you know that chromosome structure and transposition can be, well, a total nightmare. But don't you worry, because today we are going to turn these super tricky topics into your highest scoring sections. All right, let's get right into it. I mean, seriously, just think about that for a second. 6 feet of DNA has to get crammed into a microscopic cell. It sounds completely impossible, right? Well, this single question is the ultimate challenge of genetics. And everything we're about to cover, from supercoiling to these crazy jumping genes, is nature's incredibly clever solution to this gigantic packaging problem. So here's our game plan for today. We're going to start with the beautiful, simple design of bacterial DNA. Then we'll level up to the much more complex eukaryotic genomes. After that, we'll dive into the wild world of jumping genes, and we'll wrap it all up with a quick-fire checklist to make sure you've got this stuff locked down for your exam.
Okay, first up, bacteria. We're starting here because it's the perfect foundational system for understanding how any kind of DNA that's organized. It's the OG model.
What's so cool about the bacterial chromosome is just how efficient it is. Take a look. It's just a single circular loop of DNA. You've got one single origin of replication to start the copying process. You have all the genes that are the blueprints for proteins, and then these little intergenic regions in between. It's just a masterclass in minimalist functional design. 1,000 times. That is just an insane level of compression. So how in the world does a bacterium pull this off? Well, it relies on a really powerful two-step process. Step 1 is all about forming what we call loop domains. The best way to picture this is to imagine the DNA blooming out from a central point, kind of like petals on a flower, and they're all held in place by special proteins. But even that isn't enough to get it small enough. So for the next level of packing, that entire flower structure gets twisted up on itself. And this, my friends, is called DNA supercoiling. So supercoiling is basically creating coils on top of the existing coils. And this brings us to a huge exam term, topoisomers. You have to know this one. Topoisomers are just DNA molecules that have the exact same sequence, but they are twisted differently. That's it. Same DNA, different twist.
And this image shows you the two ways you can do it. If you start with your relaxed DNA in the middle and you twist it even tighter or overwind it, you get what's called a positive supercoil. But if you twist it the other way and underwind it, you create a negative supercoil. Now, for bacteria, one of these is way more important.
And here it is, your big, high-yield exam tip. Write this down. Bacterial DNA is kept in a negatively supercoiled state. So why does that matter? Well, this underwinding creates a kind of stored tension in the molecule. It's like a coiled-up spring just waiting to pop open. And that makes it so much easier for the cell to pull the two DNA strands apart, which is absolutely critical for both copying the DNA and for reading the genes. Now, the cell maintains this perfect balancing act using two enzymes that are essentially in a tug of war. You've got DNA gyrase, which is a type of topoisomerase 2, and its job is to burn energy, to burn ATP, to introduce those important negative supercoils. Then you have its rival, topoisomerase 1, which does the exact opposite. It relaxes the negative supercoils. This constant push and pull keeps the chromosome right in that functional sweet spot. And this isn't just some abstract concept. This has real-world life-or-death consequences. Because DNA gyrase is so essential for bacteria, it makes a fantastic target for antibiotics. Drugs like Cipro work by specifically blocking the bacterial DNA gyrase. This stops the cell from managing its DNA, the chromosome basically becomes a tangled mess, replication grinds to a halt, and the bacteria die. It's pretty genius, actually. Okay, so we have totally nailed the bacterial system. Now, let's level up. It's time to tackle the eukaryotic genome, which is a massive upgrade in pretty much every way. Size, complexity, you name it.
This slide right here is perfect for your notes. Seriously, screenshot this. What are the key differences? Well, our DNA is split into multiple linear chromosomes, and they're all tucked away safely inside a nucleus. We also have many origins of replication to copy our huge genomes efficiently. And our genes are usually broken up by these non-coding bits called introns.
Now, here's something that might surprise you. You might think a bigger genome automatically means a more complex organism, right? Well, that's not always true. I mean, look at the salamander. Its genome is absolutely enormous, way bigger than a human's. And it's not because it has more genes, it's because its DNA is stuffed with these huge, long stretches of repetitive sequences. And guess what? We're not all that different. Just look at our own genome. A measly 2% of our DNA actually codes for proteins. 2%. So what's the rest? Well, a whopping 59% of it is made of repetitive DNA, which of course begs the question, what is all that other stuff doing? Well, a huge part of it is made up of these things called jumping genes. And that brings us to our next big topic, transposition. This is the idea that our genome isn't some static, boring blueprint. Oh no, it's a dynamic, shifting landscape where pieces of DNA can literally get up and move around. So the official definition is that transposition is the movement of a DNA segment, we call it a transposable element, or TE, from one spot in the genome to a totally new one. And yeah, these are the famous jumping genes that were discovered by the amazing Barbara McClintock. Now, these TEs move around in two main ways. The first is called simple transposition. You can think of this as cut and paste. An enzyme called transposase literally snips the element out of its spot and pastes it somewhere else. The other way is retrotransposition, which is more like copy and paste. Here, the element is first copied into an RNA molecule. Then, an enzyme uses that RNA to make a brand new DNA copy, which then gets inserted somewhere else. So the original element stays put, and the genome gets a new copy. Okay, everybody lean in. This is a massive exam trap, and it trips up so many students. You have to know the difference between two types of repeats. First, you have inverted repeats, or IRs. These are part of the jumping gene itself. They act like bookends. Then you have direct repeats, or DRs. These are not part of the element. They are created in the host DNA during the actual moment of insertion. This diagram makes it perfectly clear. See the inverted repeats, the IRs? They're inside that yellow box, part of the transposon itself, pointing towards each other. Now look at the direct repeats, the DRs. They're outside the yellow box, flanking the entire thing. They were made when the element jumped in. Getting this difference down is absolutely crucial. Phew, okay, we have covered a ton of ground. Now it's time to make sure it all sticks. We're gonna run through a quick, high-yield checklist. So I'll ask the question, you hit pause, and really try to answer it out loud. Ready? Let's start with chromosome structure. First question, what are the two main ways bacteria pack their DNA? Did you get loop domains and supercoiling? Awesome. Next, why is negative supercoiling so important? The key is that it creates tension to help separate the DNA strands. All right, what enzyme is the target of Cipro? That's right, it's DNA gyrase. And finally, name 3 key features of a eukaryotic chromosome. You could have said linear, multiple origins of replication, or that they're found in a nucleus. Now, let's do transposition. First, explain the difference between cut and paste and copy and paste. Cut and paste is simple transposition, and copy and paste, or retrotransposition, increases the copy number. Okay, what enzyme do you need for simple transposition? That would be transposase. And now for the big one, what is the difference between an inverted repeat and a direct repeat? Remember, IRs are part of the element, while DRs are created in the target DNA during insertion. And that really brings us to our final thought for today. The discovery that bits of our DNA can just move completely changed how we think about our own genetics. It's not this fixed, unchanging blueprint we once thought it was. It's much more like a dynamic, living document that's constantly being edited and revised. So, is it a stable blueprint or a living document? Something to think about. Thanks so much for tuning in.